The COVID-19 Immunity Task Force and CanCOVID have partnered on a monthly event series, Research Results and Implications, to offer researchers, policymakers, and Canadians with the latest COVID-19 related results. To learn more about how both organizations have been helping to inform public health policy during the COVID-19 pandemic, please visit our websites at covid19immunitytaskforce.ca and cancovid.ca.
Okay. Good afternoon and good morning to everyone. I'm Dr. Tim Evans, one of today's speakers. Bonjour, je suis Dr. Tim Evans, l'un des orateurs aujourd'hui. I've been asked to share some brief instructions for our French speaking attendees so they can access the live translations, and then I will resume in English. On m'a demandé de partager quelques brèves instructions pour nos participants français afin qu'ils puissent accéder aux traductions en direct. Note à l'attention de nos participants francophones, nous vous invitons à vous rendre au bas de votre écran Zoom où se trouve une icône ressemblant à une globe. Pour indiquer votre langue de préférence, cliquez sur ce globe et sélectionnez French. Dès que notre conférencier commence à parler, vous allez entendre notre traducteur. En tout temps, si vous souhaitez revenir à la version anglaise, revenir à la version anglaise, vous n'avez qu'à cliquer sur le globe et à sélectionner Off. De plus, vous verrez qu'une version française de la présentation a été partagée dans le Q&A. Si vous désirez suivre la présentation française uniquement. Tous nos présenteurs aujourd'hui présenteront en anglais. We'll take a moment now to allow our French attendees to turn on their live translation service. As I indicated on the globe below, before I introduce our moderator. So it is my pleasure to introduce our moderator today, Dr. Nazeem Muhajarine. Dr. Muhajarine is a professor in community health and epidemiology at the University of Saskatchewan. He's the lead in public health systems and social policy pillar at COVARNet and the director of the Saskatchewan Population Health and Research Unit, as well as a theme lead advisor at CanCOVID. We're grateful that he's been able to lead us in today's event. Dr. Muhajarine, thanks for being here today, and over to you. I think, Doctor, yes, you're on mute.
Sorry, I hope that is the only time that I have buttery fingers here. Thank you, Dr. Evans, for your kind introduction and for your help in setting up our French attendees with the live translation. Welcome, everyone, to CITF and CanCOVID's fourth installment in their seminar series, Research Results and Implications. Our topic today is, how long does immunity to COVID-19 last? Waning immunity, boosters, and dosing intervals. Before we proceed, I want to pay respects to the first inhabitants of this land and their cultures. For example, I'm joining you from Treaty 6 territory and the homeland of the Métis people in Saskatoon. Before we begin our presentations, I'd like to point out to our attendees that you don't have to wait until the end of today's session to ask your questions. If you type them into the Q&A at the end of the presentation. A quick word of the format. We have 5 presentations starting and ending with Dr. Evans. I will introduce each speaker before they speak. Well, let's begin. I would like to invite back our first speaker of the day, Dr. Timothy Evans. Dr. Evans is the Executive Director at CITF, Canadian Immunity Task Force. He'll be sharing a brief overview of the CITF's area of research and various studies. Dr. Evans, The mic is yours.
I'm sorry to interrupt. Dr. Evans, if you could unmute yourself, please.
Thank you. Sorry, it's infectious. The COVID-19 Immunity Task Force mandate was provided by the Government of Canada in April 2020. And that mandate was to catalyze, support, fund, and harmonize knowledge on SARS-CoV-2 immunity for federal, provincial, and territorial decision makers to inform their efforts to protect Canadians and minimize the impact of the COVID-19 pandemic. Next. The CITF has a number of priority research areas that are illustrated on this slide, and it ranges from seroprevalence studies that assess the extent of SARS-CoV-2 infection in Canada, to understanding the nature of immunity arising from infection in terms of immune science, the development of tests to understand levels of immunity, promoting vaccine surveillance to monitor effectiveness and safety of vaccines, understand the need for boosters and the timing of those boosters in different populations, zero in on the children across Canada and understanding research safety and in understanding safety and effectiveness and immunogenicity of vaccines, and also modeling trends in overall immunity in Canada. Next. We have about 111 studies that are active across the country at this point in time, across all of those areas of research. Next. And I think for this session in particular, we're going to focus in on vaccines and the immunity that arises from vaccines, recognizing that it's a critical and necessary resource to protect Canadians from COVID-19. This reflects the fact that up until November 2021, levels of infection-acquired immunity across Canada were very low. That's testimony to good public health. But once vaccines became available in 2020, and knowing that we needed vaccine acquired immunity to protect ourselves from the virus, we've had a massive rollout in the last year, which has seen us go from virtually no vaccine acquired immunity to very high levels of vaccine acquired immunity. And we've seen now through multiple studies, not only here in Canada, but elsewhere, that vaccines have delivered as promised. They have prevented thousands of hospitalizations and death, and have, in many respects, decreased infection and transmission significantly. And so I think also the way in which vaccines were rolled out across Canada, both the scale and the longer dosing intervals between first and second vaccines, have also contributed very positively to population immune protection. Next slide. So this is just an example of the extent to which the hospitalizations and deaths have been limited or largely concentrated in the unvaccinated compared to the partially and fully vaccinated populations. Next. However, and as we'll hear today from some of these CITF supported studies, antibody wane is real, and vaccine surveillance has identified some very concerning trends. Wane in antibodies, especially in older age groups and immunocompromised populations, has been noted now since late August. New Omicron variant associated the new Omicron variant is associated with very high levels of breakthrough infections, or much higher than the previous variants. And we're seeing transmission despite full vaccination in many people. So this presentation today is going to focus on the evidence on waning antibodies from our CITF supported studies. And we look forward to our presenters giving us more details on their work. Just to mention that the results are being shared in real time, and it's a mix of both published and unpublished material. Some have gone through peer review, others not. And when we discuss Omicron, you can be sure results have not gone through peer review, as we've only been dealing with this variant for just a bit over a month. So with that, I'll hand it back to you, Dr. Muhajarine. Thank you.
Thank you, Dr. Evans, for providing that overview of CITF and the work your team is doing there. Our next speaker is Dr. Jeff Kwong, a senior scientist at ICS, Institute for Clinical Evaluative Studies, a scientist at Public Health Ontario, and a professor at the University of Toronto. He'll be presenting on the effectiveness of COVID-19 vaccines over time in Ontario. Dr. Kwong, welcome. The floor is yours.
Great. Thank you so much for the introduction. Can I have the next slide, please? So I just wanted to say that I have no conflicts of interest to declare related to this work. Next slide. So, I think there's no question, there's many studies that have found that vaccine effectiveness declines over time. Some studies, some in the US have suggested that there seems to be lower vaccine effectiveness against the Delta variant compared to the Alpha variant. The problem with analyses like that is that in the US, and as in other countries where they had a lot of rapid uptake initially, in a relatively short period of time, the people that got exposed to Delta, had a longer time since their second dose than those who were exposed to Alpha. So it's hard to tease out whether there's differences in vaccine effectiveness against different variants, as opposed to just when those variants appeared in time. And if later variants, people would have had more waning of vaccine effectiveness, then it'll appear that there's lower vaccine effectiveness against a particular variant. Now, there have been some Canadian studies. So there's a preprint from BC and with data from BC and Quebec that suggests that longer intervals between the first and second dose leads to higher vaccine effectiveness. Now, one issue is that people who've had a longer interval between doses would have less time for follow-up after their second dose. So it's perhaps people with longer intervals haven't had as much time for waning to occur as compared to people who've had shorter intervals between doses. So evaluating waning of vaccine effectiveness in Canada is challenging because there have been many changes, not just in the circulating lineages of SARS-CoV-2, but also changes in the vaccine program policies. So because we've had vaccine supply constraints, we've had to have changes in the dosing intervals. So that's been, all of these things are challenging in evaluating waning of vaccine effectiveness. Next slide, please. So the objective of our first study was to estimate a vaccine effectiveness against multiple outcomes over time while accounting for changes in the circulating SARS-CoV-2 lineages and dosing intervals. Next slide. So I'll talk briefly about the test negative design. This is the design that we used. It's very similar to a nested case control study, and this is adapted from use for influenza vaccine effectiveness studies. So what you do is you take people who present with symptoms and you swab them to see if they have the virus. Those who test positive, you treat as cases, and those who test negative, you treat as controls. And for each group, you look at the proportion of that group who have been vaccinated, and then you can calculate an odds ratio. So the odds ratio is just the odds of vaccination in those who test positive, or the cases, divided by the odds of vaccination in the test negatives, or the controls. And then you can calculate vaccine effectiveness as 1 minus the odds ratio, and that's how you get the vaccine effectiveness. So next slide, please. So what we did was we used linked databases in Ontario, and we restricted the study population to those who are age 16 or older based on initial eligibility for vaccines, and those who are community dwelling. So we excluded residents of long-term care homes. And we essentially did kind of like 3 mini test negative studies where we had 3 different periods. So the first period was basically January through March and a little bit of April. So that was a mixture of the original Wuhan strain and when Alpha started circulating. Then we had what we're calling the Alpha period, which is basically April till the end of June. About 77% of specimens during that time were the Alpha variant. And then we had the Delta period, which was basically July till the end of October. And so 97% of specimens during that time were Delta. We looked at 3 different outcomes. So the first outcome was any infection. So it's infection irrespective of symptoms or severity. So if you just tested positive for SARS-CoV-2, you were considered a case. And that was in anyone who went for testing. And in the second, for the second outcome, we looked at symptomatic infection. So those are people who presented for testing, and there was a record there was indication that they had at least one relevant SARS-CoV-2 symptom that was recorded in the database of the Ontario Laboratories Information System database, that they had at least one symptom at the time of testing. And then lastly, we looked at severe outcomes, which was defined as either hospitalization or death from COVID. So it was either they were recorded as due to COVID, or it could have been shortly after a positive test. Okay, so next slide, please. For the exposures, we looked at the time since their second dose, and we considered anyone who had any combination of 2 mRNA vaccines. So they could have been Pfizer, or Moderna, or it could have been Pfizer, Moderna, or the vice versa. And then we also looked at the interval between doses. So we looked at whether it was a short interval, less than 5 weeks, between 5 and 7 weeks, or longer than, or 8 weeks or longer in between the first and second doses. For the covariates, we've considered the demographics such as age, sex, and geographic region. So it's based on a public health unit and grouped into larger regions. Next, we've also considered the calendar time. So the date of their test divided into 2-week periods. We also looked at the number of SARS-CoV-2 tests that they had prior to the start of the vaccination program. So that's a proxy to identify healthcare workers. And we also looked at a history of influenza vaccination, and that's a proxy for health behaviors of the individual. We considered a number of comorbidities that increase your chance of severe COVID infection. And then we also looked at 4 area level measures. They included neighborhood income, the proportion of the population who are essential workers, so people who generally can't work from home, the average household size, the number of persons per household, and then the proportion of the population who self-report as visible minorities. Next slide. So what we've got here are VE estimates over time. So you can see in the graph on the right, on the x-axis, we've got time in days since the second dose, also expressed as months. And so you can see we go all the way up till about 8 months after the second dose. On the y-axis is the vaccine effectiveness. And so you can see we've got 3 lines. In the red line is hospitalization or death or severe outcomes. The next one in orange is symptomatic infection. And then third line in the blue is infection. So that's any infection. So you can see here that vaccine effectiveness is consistently higher against severe outcomes than either symptomatic infection and infection. You can see that the vaccine effectiveness against severe outcomes remains very high, above 90% all the way through. Whereas for infection, you can see that vaccine effectiveness declined from about 90% to about 73% by 8 months or more after the second dose. And so, so that's what you can see there. Next slide, please. And comparing the estimates using the test negative design with randomized controlled trials, I've just presented here the data from the Pfizer and the Moderna clinical trials. So those are the solid lines where they had data up till 6 months. And so you can see that the red line is for Moderna and the blue line is for the Pfizer. And then you can see in the dashed lines the estimates from the test negative design for symptomatic infection, which most closely aligns with the outcome used in the clinical trials. And you can see that the dashed lines, pretty closely align with the solid lines up till the 4 to 5 month mark. And you can see the differences between the red dashed line and the red solid line, the blue dashed line, the blue solid line are less than 5%. And then in the test negative design, we can go onwards to 6 to 7 months and then 8 months or beyond. Now you'll see that it appears that the vaccine effectiveness for the Moderna vaccine is a lot lower than the Pfizer vaccine at the 8-month mark. And I suspect that may be due to differences in who received those two vaccines in that very early period of the vaccination program. So these were people who got their second dose in January, maybe February. And so, at that time, it was mostly healthcare workers who were getting the Pfizer vaccine, whereas the people who got Moderna were mostly residents of retirement homes. So they're not long-term care facilities, but retirement homes, which are not considered long-term care facilities, were included in these studies. Next slide, please. So now what we've done is we've stratified the groups further, not just by the period of test, which is a proxy for the SARS-CoV-2 lineage, but also by dosing interval. So we've got multiple lines here in different colors, and then also solid versus dashed. So we'll focus first on the solid lines. And this is for VE against infection amongst those who are aged 16 to 69. And we've got corresponding plots for 70 and older, and also against other outcomes. But I'm showing this one only just in the interest of time, and because this had the largest sample size. So focusing only on the solid lines, you can see that the difference between the red, yellow, and blue, so those are the differences between the dosing intervals, was actually quite minimal, less than 5%, suggesting that people who had a shorter dosing interval had pretty similar vaccine effectiveness as with a longer dosing interval. You can see that the blue line is actually on top of the orange line. And then the orange line is not very far off from the red line. There's a little bit of separation between the red line and the orange line. Now, comparing across across the variants, you can see that the red solid line and the red dashed line, that's comparing Delta versus Alpha, are also very similar. And even the very dashed line, the pre-alpha is also quite similar. So there isn't a huge difference between the variant in terms of the effectiveness of, in preventing infection. So I think the bottom line here is that it appears that the time since the second dose has the greatest impact on VE compared to looking at SARS-CoV-2 lineage or dosing interval. Next slide. So moving on to looking at VE against Omicron, this was, we did some work over the holidays and we posted it on medRxiv. So it was posted on January 1, where we looked at the vaccine effectiveness against Omicron. And so we restricted this study to those who are 18 years or older because those who are under 18 were not eligible to get boosters. And we also restricted to community dwelling individuals. And so we looked at people who were tested for SARS-CoV-2 between November 22nd and December 19th. So that was the first 4 weeks after identification of Omicron in Ontario. So the first case of Omicron was identified in Ontario on November 22nd. And because we didn't have access to the data on symptoms at that time for this latest data set, we had to use infection as our outcome. So we looked at infection irrespective of symptoms or severity. And then to classify cases as Omicron or Delta, we used results of whole genome sequencing or the presence of the S gene target. So the S gene target, if it's not present, then that suggests that it's Omicron. And if their S gene target comes back positive, then we consider them as Delta. So S gene target failure positive means the absence of the S gene target. We call that Omicron. And S gene target SGTF negative, then we call those Delta. And then we also looked at it by the date of test. So prior to December 3rd, there were less than 5% were Omicron at that point, so we considered those cases as Delta. So next slide. And so on the right-hand side, you can see on the x-axis, we've got days since the second dose for the first 5 time points. And then after that, on the right-hand side of the graph, you can see days since the third dose. And we've got first any mRNA, and then we've also split up into the boosters of Pfizer and Moderna as boosters. So what we considered here was people who had received at least one mRNA vaccine for the first two doses of the primary series. So this allowed us to include people who received AstraZeneca and Pfizer, or AstraZeneca and Moderna, because prior studies have shown that people who received at least one mRNA for the primary series have a similar VE as those who received 2 mRNA doses. And so in the black dots, you can see VE against Delta is very high. But it does decline over time after the second dose. So it declined to about 71%, by about 8 months after the second dose. And then after the booster, after the third dose, it climbed to 93%, 7 days or more after the third dose. Whereas infection against Omicron, you can see that the vaccine effectiveness is a lot lower than against Delta. And it only after the booster, it goes to 37%. And you can even see after the 2 doses, there are a few estimates that are below 0, suggesting that there's negative vaccine effectiveness. Okay, next slide. So since then, we have done some further analyses, and these are going to be posted on medRxiv soon. So once again, we had the population of age 18 years or older. And in these new analyses, we restricted to those who are tested from December 6th to December 26th. So that's weeks 3, 4, and 5 after first identification of Omicron in Ontario. And the reason for this is that was the period that there was universal testing for SGTF in Ontario. So all specimens were supposed to be undergo testing for the SGTF, and that allowed us to be as specific as possible in identifying which ones are Omicron and which ones are Delta. And now in January, we had available to us the information on symptoms. So we use symptomatic infection for this analysis. And then we also looked at severe outcomes, so hospitalization or death. And once again, we classify cases Omicron or Delta based on sequencing or SGTF, and we didn't use any dates because some of them where the specimens either have no SGTF information or inconclusive, those cases were excluded. The next slide, please. So here we have VE against symptomatic infection, and you'll note that the x-axis is the same as pre-4, but the y-axis is different. And so you can see once again against symptomatic infection, VE, against Delta is actually, it declined to 80% by 8 months after the second dose, and then recovered to 97% after the third dose. Whereas VE against symptomatic infection caused by Omicron is far lower against Delta after the second dose. And you can see that it, starts off below 40, and then it, quickly drops off to below 20. And then by about 6 months, it's basically 0. And then after the third dose, vaccine effectiveness does increase up to about 60%. And so these estimates are more closely aligned to estimates seen in other countries. And this difference was largely because of using a different outcome. So using symptomatic infection as opposed to infection. Next slide. And then you can see here against severe outcomes that the estimates against Omicron, so first I'll start with the estimates against Delta. So they're very high after 2 doses, even up to 95% after 8 months, and it goes up to 99% after the third dose. And then for Omicron, you can see that the estimates are quite imprecise. The ones that do not cross 0 are in the range of 82, at about 6 to 7 months, and then even at 8 months, the point estimates at 86, but with a very wide confidence interval. But after the third dose, vaccine effectiveness is 95%, so very similar to Delta against the severe outcomes with 3 doses. Next slide. So here what we've done is we've we've analyzed the data where we progressively add weeks. So we start back with like the people, the cases from November 22nd, and then up to December 12th. So that's weeks 1, 2, and 3. And then we add week 4 in the middle plot. So that's up to December 19th. And then we add the 5th week up to December 26th on the last plot on the right. And so you can see that the VE estimates are very imprecise in the left-hand side. And then as you move more to the right, they become much more precise by much narrower confidence intervals. The point estimates do go from negative at the 180 and the 240 plus days on the left-hand side, and then they do go to around 0 so it shows that in the early days there was some bias, and this likely was because the people who were getting Omicron were those who were double vaccinated because they were travelers, and you had to be double vaccinated to travel, or you were household contacts or social contacts of these travelers, and you were probably double vaccinated as well. So basically, in the early days, a lot of the unvaccinated people didn't have the opportunity to be exposed to Omicron. And that's why, one of the reasons why it looks like you get negative VE estimates at the beginning, although the use of the outcome measure was also a big factor as well. Okay, next slide. So basically, these are just some of our learnings. The choice of the outcome measure is actually very important. So VE against infection is more susceptible to downward bias than symptomatic infection and severe outcomes. And this has to do with indications for testing. Why are people getting tested? If you're asymptomatic, is it because people who are vaccinated are more likely to get tested because they had, let's say, exposure, or they're just more like they have a greater propensity to go get tested, and then they're more likely to be picked up, and then it looks like VE is worse because of that outcome. The second point is about the study period. So estimating VE in the initial weeks following Omicron circulation is also susceptible to downward bias. And that's because, as I mentioned, because of differential exposure to Omicron in vaccinated versus unvaccinated populations. In Ontario, we had vaccine certificates. So people who were vaccinated were probably more likely to get exposed. And it took a few weeks for Omicron to diffuse through the population and to reach unvaccinated individuals. The third point is about how to classify Omicron versus Delta. So using dates to define Omicron is actually leads you to be susceptible to upward bias in the VE estimates. So there are some times when we've seen that more of the unvaccinated people had Delta. And so if you classify those as Delta instead of Omicron, then you'll actually get higher VE estimates than the truth. So all of these are factors in the decisions that we made in our latest analyses. And then in the background of all of this, there's challenges in terms of access to PCR testing and also availability of whole genome sequencing and SGTF screening, which varied over time. And so, this is getting really hard now with people using the rapid antigen tests and not being able to get PCR tests. On December 30th, the Ontario government restricted PCR testing to very selected groups. And even prior to that, it was getting harder and harder for people to access PCR testing. Next slide. So in summary, we see that VE wanes steadily over time following the second dose, and it appears to be more so against infection than symptomatic infection, and then against severe outcomes. And the time since the second dose, at least up to 8 months, so maybe over long term, this may not be the case, but at least over this relatively short period of time of 8 months, it's, time since the second dose is a more important determinant of vaccine effectiveness than either the lineage, at least pre-Omicron, or dosing interval. For symptomatic infection, VE against Omicron is lower than against Delta, and whether that's after 2 or 3 doses. But for severe outcomes, VE against Omicron appears to be comparable to Delta, at least after the 3rd dose. And I'll just leave with one last point, is that estimating VE against Omicron is very challenging because of the things I've mentioned. You introduce a new variant that has immune evasion in a highly vaccinated population. There's kind of differential exposure in the vaccinated and unvaccinated populations. differential access to testing, and then being able to classify which ones are Omicron versus Delta has also been challenging. So lots of issues that we've come across in doing these analyses. Thank you very much for your attention. And just wanted to acknowledge the many people. This is the Ontario study team of the Provincial Collaborative Network of the Canadian Immunization Research Network. And next slide, the funding from CITF and from CERN via CIHR and Public Health Agency of Canada. Thank you very much.
Thank you very much, Dr. Kwong, for sharing your team's work on estimating vaccine effectiveness against multiple outcomes over time, including, very importantly, that updated data on Omicron. Really, really interesting, very timely. I'm sure there'll be questions. So thank you very much. Now it is my pleasure to introduce our next speakers who will be co-presenting on the COVID-19 vaccinations and infections in long-term care in Ontario. Dr. Dawn Bowdish is a professor at McMaster University and Canada Research Chair in Aging and Immunity. She will be joined by Dr. Andrew Costa, an associate professor and Cellular Research Chair in Clinical Epidemiology and Aging at McMaster University. So the floor is yours, Dr. Bowdish and Dr. Costa.
Thank you so much. It's my honor to present on behalf of my partner, Dr. Costa, our partners, our various long-term care homes, and our research team. Next slide, please. Neither Dr. Costa or I have any conflicts of interest to declare. So in our study, where we're looking at vaccine immunogenicity in people who are living in long-term care and retirement homes, we'll be taking and showing you measures of both antibody quantity and antibody quality. So on the left, what you're seeing is a picture of the virus we've all grown to hate. And as you know, the spike protein, which is highlighted here in red, is mimicked, is replicated in the vaccines that we have. And in fact, we measure antibodies towards the spike protein as proof that people have responded to the vaccine. Now, if we were to go even further, blow up even further in this, we would see that the receptor binding domain is a small domain within the spike protein, which historically we've measured antibodies against, because those antibodies are the most effective at binding to it and preventing the virus from getting into us. However, in the context of Omicron, we may have to revise the relative importance of these two different kinds of antibodies. And I'll go into that in a second. One of the most important measures of quality is the ability of the antibody to neutralize the virus. So for example, if the antibodies you make a lot of antibodies, but they don't stick to the virus, they won't be able to keep the virus out of us. So on the left, we have the perfect scenario where a person has made strong neutralizing antibodies that can stop the virus from getting in. And on the right hand is the suboptimal situation where the person might be making antibodies to the virus, but they may not be either sticky enough or there may not be enough of them to prevent the virus getting into the cells. So the antibody measures you'll see me presenting are antibodies to the spike protein, that receptor binding domain, and also these neutralizing antibodies that can stop the virus from getting into the cells. Like I said, with Omicron, we may have to revise what we think is the most important measure of immunogenicity. When we were looking at the original strain, we used to measure immune responsiveness by looking at the spike antibodies, but to be perfectly frank, the antibodies I was most interested in were the ones that bind that receptor binding domain, that one specific domain that stops the virus from binding our cells. Omicron's acquired dozens of mutations. The vast majority of them are in that receptor binding domain. And a very slight change in the shape of that means that fewer of these RBD antibodies will bind to that area. And instead, we may need to start thinking about having a lot of these antibodies, having high quantities of antibodies to prevent that binding and neutralization. And I'll show you our data suggesting that this third dose has done just that. Next slide, please. We have over 17 partner homes and incredibly committed partners across Ontario, all of whom have donated blood to our study. Next slide, please. But I'd like to pause for a second and reflect on the incredible people we're doing this for. The youngest residents in our study of long-term care are in their 40s, the oldest are over 100. And their families have been kind enough in many cases to enroll people in our studies so we can get them the answers that they need to keep themselves safe. And so we're incredibly grateful and need to acknowledge our incredible partnerships. Here's the first bit of data. So these data that I'm showing you now are prior to the third dose. And on the left hand, what we're looking at are those anti-spike antibodies. On the right hand, we're looking at the RBD antibodies. And you'll see this common color coding throughout the talk, where the red zone means that those antibodies are falling below the level of detection. Effectively, people have none of these antibodies. The green zone is sort of a peak immune response. And so the comparator groups that you can look for on the right of the graph are a group of healthcare workers who are younger and generally healthy and fit, and then in some cases, people who've recovered from infections. So what you see is that after the second dose, there is a decline. Those blue bars are the median values. So about half the people are above and half the people are below. And what you can see is that although the spike antibodies wane more slowly, those RBD antibodies, the ones that are so essential to bind to that specific region, are declining very quickly. And by 6 to 8 months, we saw that fully half of our participants were in that red zone. And I remember Andrew and I looking at these data and being really, really upset because we were so worried that our research participants might not be protected without a third dose. Next slide, please. Also, if we look at this neutralizing antibodies, the ability to prevent these from getting in, again, the blue line representing the median value, half the people are above or below, these seem to decline even faster, where by 4 to 6 months after that second dose, we were seeing that fully half of our participants were below that level of detection, and we would not predict that they would be protected from infection. Next slide, please. So fortunately, we were able to make the recommendation that a third dose was required, and honestly, it looked like it happened just in time. Cases in long-term care were beginning to creep up with the Delta variant, but those third doses came in, and with weeks, many people in long-term care and virtually all our research participants were protected by a third dose. And we saw a steep decline in breakthrough infections, including serious infections in this population. Of course, this was against Delta. And if we look at the next slide, we'll see what Omicron's done to us. So we saw this wonderful decline, and we had a brief period of peace of mind in October and November when rates were fairly low. But as we've heard, that Omicron is an immune evasive variant. So many of our participants who don't make the highest quality antibody, either because of their age, their complex medical conditions, or the medications they are on, are of course vulnerable to breakthrough infections. And that's exactly what we're seeing. So why do we give 3 doses? Well, 3 doses this is in every textbook of immunology. And the reason for 3 doses is that if we look at the next slide, we'll see that it gives basically antibody quality as well as quantity and cellular immunity time to really mature and make fully formed immune response. Next dose, next slide, please. Also next dose, please, as you will soon see. Now, these were some data that we were able to do by looking at the rate of decline of some of these responses after the second dose and the third dose. So here what we're looking at are those RBD antibodies against that receptor binding domain. In the lilac color, you're seeing how that rate of decline after the second dose in our long-term care participants. And if you compare that to the dark blue, you see they got that third dose and they had this incredibly high response, which was probably contributed to some of that protection. However, if we start to model that rate of decline, it doesn't look like it's any different than after the second dose. Now, by this point, we had started to recruit retirement home participants who would be, in general, healthier older adults. And again, you saw a very steep decline over time of those antibodies. You saw an incredible increase after that third dose, but there does seem to be a rate of decline. And this will be even more obvious on the next slide when we look at the neutralizing antibodies. So on the left, we're looking at neutralizing antibodies to the wild type strain, the strain we were all immunized against. And again, you see that there's a nice response after that second dose in both long-term care and retirement that levels off, a beautiful jump after that third dose. But again, if we were to model the rate of decline, we would see that it would be no different. What you're looking on the right is the beta variant, which until Omicron was the most immune evasive strain. And you can see that those rates are, those doses are lower, or those antibodies are lower, but we still see that decline after the second dose. So based on these data, Andrew and I recommended that we should we should have a fourth dose recommendation for these populations, because despite the fact they are able to get up nice and high, they decline very quickly. And if we were to make some predictions and model it out, we would have predicted that would be around the 3-month mark. Next slide, please. Vaccine types matters, as we've already heard. To orient you here, what you're looking at when you see a P is a Pfizer. So on the very left, we're seeing people, their antibody levels after 2 doses of Pfizer. And then we see after their third dose, so Pfizer, 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 you can see that we get this wonderful jump in the spike antibodies. We also see that people who had Pfizer, Pfizer followed by Moderna have this lovely increase up until what we call the green zone. Then we see people after their second dose of Moderna, and you can see that those levels are higher than those after their second dose of the Pfizer. And again, if they get a third dose that is Moderna or Pfizer, it's up nice and high. Let's look at the next slide. And these are those RBD antibodies, those ones that had declined and given Andrew and I such a poor night's sleep. You can see after the second dose of Pfizer, almost half our participants were in the red zone, but giving them that third dose brought them up. And if that third dose happened to be Moderna, it would be even higher still. After 2 doses of Moderna, they looked like they had more of these antibodies than after 2 doses of Pfizer, and those jumped right back up with very few of our participants in the red zone after that third dose. Next slide, please. And if we look at the neutralization against that immune evasive beta version, on the left-hand side, we see 2 doses of Pfizer versus 2 doses of Moderna. And disturbingly, those neutralizing antibodies were very, very low, but they do increase very substantially after that third dose. And we can use this as a proxy to predict how well they would be protected against the Omicron, which is also an immune evasive version. So third doses are essential, and I'm certain fourth doses will be as well. Next slide, please. There's also some good news here, and I so rarely get to give good news in my line of work, so I'm hoping that you will allow me to relish this moment. We've, in our study, been looking at both antibody responses, which we believe are key to susceptibility to infection, do you get infected or not, and also to transmission. There's emerging data that the cellular responses to our immune response may be what end up protecting us from severe infection, hospitalization, and death. And cellular responses against our vaccine seem to be more conserved against Omicron than the antibody responses. Next slide, please. In general, older adults have a problem with cellular immunity. They have fewer immune cells to give or to donate to the cause, and oftentimes it's a challenge to continue boosting those cellular immune levels. So on the left-hand side, if you look at our participants after their post-2 and post-3 dose, you see that there is an increase in these spike-specific T cells that are dedicated to fighting this infection. And in fact, after the third dose, we have fewer of them in the red zone. And when we look at paired samples, so the same person after their second dose and after their third dose, we see that those do seem to go up in almost everybody. Again, Moderna seemed to be more effective than Pfizer in inducing a cellular immune response. And we hope and remain cautiously optimistic that good cellular immune responses may contributing to reducing the severity of poor outcomes in this very, very vulnerable population. Next slide, please. To bring it all home, there is, in fact, very good evidence that there was a substantive improvement in immune responses after the third dose of the mRNA vaccine. Our recommendation has been that Moderna is the more immunogenic in this population, and many homes in Ontario have adopted this recommendation. Our data also supports that older adults in retirement homes and frail older adults in other congregate settings will benefit from having a fourth dose, which we expect to provide the best humoral and cellular immunity against Omicron. Next slide, please. I have many, many, many people to thank in this case. Our immunology team, including Dr. Jessica Bresnett and Ali Zhang, our data analytics team who's brought these data to you together, our program manager who's able to coordinate over 1,400 blood samples being delivered to the labs, our co-investigators, or who I call the Immunology Dream Team, and of course our partner homes, the Schlegel Villages, St. Joe's Family, and Shalom Village, who've donated resources, staff, and incredible commitment to our study. Next slide, please. I also would like to acknowledge that our convalescent data came from Dr. Isaac Nassie's team, and our data on the healthcare workers comes from the timing study led by Drs. Mylan Duong and Daryl Leong. Again, I can't thank our partners enough, nor the COVID Immunity Task Force for investing in this study. With that, I'd be happy to answer any questions.
Thank you, Dr. Bowdish and Dr. Costa, who is with us today, for sharing your work with us, and including how antibodies have declined over time in long-term care residents, and really interesting data. So let's move on to our third speaker today, and that is Dr. Victor Ferreira. Dr. Ferreira is a scientific associate at the Ajmera Transplant Center in the Department of Medicine at the University Health Network. He will be speaking about the prospective evaluation of COVID-19 vaccines in transplant recipients, PREVENT study. Dr. Ferreira, welcome. The floor is yours.
Good afternoon. Bonjour. My name is Victor Ferreira, and I'm a scientific associate at the UHN Ajmera Transplant Center. I'm very excited to be here today on behalf of Dr. Dipali Kumar to share our results investigating COVID vaccine responses in immune-compromised transplant patients. Next slide, please. I have no conflicts to declare. Next slide, please. It is estimated that as many as 3% of all Canadians, a number that exceeds 1 million people, are immune compromised, which means that one or more components of their immune system are weakened or not working properly, which can increase your risk for serious infection and cancer. You can be born immune compromised, or you can acquire an immune compromised state later in life due to several illnesses, including unmanaged HIV infection, and certain types of cancers, including blood cancers like leukemias and lymphomas. You can also become immune compromised due to immunosuppressive medications used in a variety of clinical settings, including organ transplantation. Next slide, please. Our group's research efforts primarily focus on solid organ transplant recipients, or SOTR for short. SOTR typically require lifelong use of immunosuppressive medications to prevent organ rejection. These drugs impair the immune system, including T cells and B cells, which are important for fighting infections as well as executing our vaccine responses. For these reasons, transplant recipients tend to have more severe infections, including severe and protracted influenza and COVID-19, and they may also have difficulty generating vaccine responses. Immune-compromised people were also excluded from all the major vaccine clinical trials, so we were left wondering to what extent COVID vaccines would work in this vulnerable population. Next slide, please. To address this gap in knowledge, we performed one of the first vaccine immunogenicity studies in organ transplant recipients. After 2 doses of the Moderna vaccine, only 34.5% of SOTR were positive for anti-RBD antibodies, and only 26.9% had detectable neutralizing antibodies. Importantly, we found that close to half of SOTR developed virus-specific polyfunctional CD4 T cells. And although we were encouraged by this cellular data, the overall conclusion of this study was that 2 doses resulted in suboptimal responses, especially when compared to the general public, where response rates tend to be quite high after 2 doses. Lower vaccine effectiveness has also been reported in the literature with data out of US transplant centers that estimate that SOTR are 82 times more likely to have a breakthrough infection and 485 times more likely to be hospitalized after 2 doses compared to the vaccinated general public. Next slide, please. So we considered whether a 3rd dose would improve vaccine responses. And to answer this question, we performed a world's first, a double-blind randomized control trial evaluating safety and immunogenicity of 3 doses in SOTR. We enrolled 120 transplant recipients for this study who had previously received 2 doses of Moderna. And participants were randomly allocated to receive a 3rd dose of Moderna or to receive a placebo. We arbitrarily set the anti-RBD cutoff higher at 100 units per mil and found that 55% of transplant recipients receiving a 3rd dose of Moderna reached this primary endpoint, but only 18% did in the placebo group. Neutralizing antibodies were also increased by 35 percentage points compared to placebo. And a 3rd dose of Moderna also increased virus-specific polyfunctional CD4 T cells by 6-fold compared to the placebo group. We shared this data with the FDA and Health Canada, who used it to guide their decision to expand 3rd doses to immune-compromised groups, including transplant recipients, a decision that had a direct impact in the lives of millions of people in North America. And we were also very excited to see these results published in the New England Journal of Medicine. Next slide, please. We also performed a follow-up secondary analysis of the randomized control trial data to evaluate neutralizing antibodies against variants of concern, namely Alpha, Beta, and Delta. We didn't know about Omicron at the time. This work was performed in collaboration with Dr. Anne-Claude Gingras and her team at the LTRI in Toronto. The image you see on this screen shows the 50% neutralization titers according to the different variants tested, which are labeled along the bottom. A 3rd of the Moderna vaccine, a 3rd dose of the Moderna vaccine increased the proportion of transplant patients with detectable neutralizing antibodies by around 30 to 35 percentage points. And this was seen across variants. However, as you can see, neutralization capacity did differ according to the variant tested. The Delta analysis is on the far right. There you can see 55% of transplant patients who received a 3rd dose mounted these Delta targeting neutralizing antibodies, while only 18% in the placebo group did. Responses were lowest against the Beta variant, but this is not surprising as it's also true for the general public. Overall, we were encouraged to see that a 3rd dose of Moderna also increased neutralizing antibodies against variants of concern, especially Delta, which was the dominant variant at the time. Next slide, please. In summary, 3 doses are likely required to complete the primary vaccine series in transplant recipients. But in general, these responses are still somewhat lower compared to the general public. A reassuring sign, though, across transplant studies, including our own, is that COVID vaccines are very safe, with organ rejection and serious adverse reactions being rarely reported. Very recently, Dr. Gingras and her group measured neutralizing antibodies against Omicron for us. Neutralizing antibodies were tested after the 3rd dose, and unfortunately, we found that fewer than 20% of SOTR had detectable neutralizing antibodies against Omicron, and these titers were generally low. As such, additional mitigation strategies for transplant recipients may still be needed, and that includes 4th doses of vaccine, which can now be booked in many provinces, as well as preventive monoclonal antibodies and perhaps even different vaccine protocols or platforms. The data I've shown you today is also from our center alone at UHN, and we hope to gain a more comprehensive picture of the safety and immunogenicity of COVID vaccines in our national collaborative research study called Prevent COVID, led by Dr. Dipali Kumar from UHN. In this study, we will measure vaccine responses in stem cell and solid organ transplant recipients, and we will look at both pediatric and adult patients as well. Next slide, please. And now for something a little different. The standard recommended interval between first and second doses of mRNA vaccine is usually about 3 to 4 weeks. But as you know, in Canada, a 4-month delay was introduced to address vaccine shortages. As part of our study, we wanted to recruit a cohort of healthcare workers from UHN because we needed a healthy control group for our studies. It just so happened that some of our healthcare workers received their second doses at a standard 3 to 6 week interval, while others had a delayed or 8 to 16 week interval between their doses. So we compared antibody and T cell responses between these two groups to see how dose interval impacted vaccine immunogenicity. And it's important to note that everyone in this study received the Pfizer vaccine. Next slide, please. On this slide is a summary of our major findings. We found that anti-RBD levels, which are shown here on the right, these anti-RBD levels were 3.2 times higher in the delayed group than in the standard interval group. However, and this is important, all participants in the study were seropositive after 2 doses, regardless of the dosing interval. In collaboration with Dr. Heidi Wood at the National Microbiology Laboratory in Winnipeg, neutralizing antibodies were also tested using a live virus assay. 50% and 90% neutralization titers were measured and were consistently found to be higher in the delayed interval group. But most of the healthcare workers were positive for neutralizing antibodies also, regardless of dosing interval and variant tested, with responses against the beta variant being the lowest. Only about 75% of healthcare workers with 2 doses responded to the beta variant. For cellular responses, we saw no differences in polyfunctional T cells between the 2 groups, but we did see lower frequencies of some CD4 T cell subsets in the delayed group, in particular, a population of CD4 T cells that produce an effector cytokine molecule called interleukin-2. Next slide, please. I would also like to share with you some new unpublished data on durability of the antibody response with standard versus delayed vaccination. These are anti-RBD antibody levels in healthcare workers at 1 and 6 months after their second dose. Green dots on the left represent the standard interval group, and the red dots on the right for the delayed group. And in this figure, there is a statistically significant decline in antibody levels by 6 months, regardless of dosing interval. But everyone was still positive at relatively high levels despite this decline. Of note, the delayed vaccine group at 6 months still had higher anti-RBD antibody levels compared to the standard vaccination group. Next slide, please. So in summary, we found better immunogenicity with delayed dosing strategies for the Pfizer vaccine, particularly with respect to anti-RBD and neutralizing antibodies. We did see some lower frequencies of CD4 T cell subsets in the delayed group, but we saw no differences in proportions of polyfunctional T cells, which are a highly activated T cell subset. At 6 months, those anti-RBD levels do decline, and we've seen some evidence in other studies of this too, but positively is mostly maintained, and antibody levels remain higher in the delayed group. So these data do support the use of delayed vaccination strategies for COVID-19. And next steps for our group include doing Omicron testing, looking at the long-term durability of the response, and looking at how a 3rd dose in the context of delayed vaccination plays a role. Next slide, please. It's been a real pleasure to speak here today, and I'd like to thank Dr. Dipali Kumar, the CITF, and Can COVID for this opportunity. I'd like to acknowledge our whole research team led by Dr. Kumar and Dr. Atul Kumar. A special thanks to Dr. Victoria Hall, our clinical coordinators, and our entire lab team of technician scientists. And I'd also like to acknowledge all of our collaborators, in particular, Dr. Kulasingham from UHN, Dr. Jingra from LTRI, and Dr. Wood from NML, along with their respective teams. Lastly, I'd like to give a special shout out to our Prevent COVID team from centers across Canada. Next slide, please. And this work was funded by CITF and the UHN Ajmer Transplant Center with contributions from the CDTRP. Thank you again for your time.
Thank you very much, Dr. Ferreira. Really interesting, and thank you again. So last but not least, I'd like to bring Dr. Timothy Evans again and ask him to sort of provide a conclusions and a bit of a summary of the results that we have heard today. Dr. Evans, the floor is yours.
Thank you very much, Dr. Muhajarine, and also to 3 excellent presentations, really beautiful. Next slide. So I think the first thing not to forget when we look at the intricate analyses that have been presented today is that in the big picture, COVID-19 vaccines have been massive lifesavers. And this you can see in the data related to the risk amongst not having vaccine protection related to infection, hospitalization, and also the number of deaths. And so I think this remains a robust finding supported by the data, and one that bears reiteration, because if we didn't have that vaccine coverage like we didn't have it just over a year ago, we would have seen a much greater strain and an overwhelming situation with respect to our hospitals. Next. So we have vaccines have prevented infection and transmission, much more so prior to Omicron. And so Dr. Kwong has shown us that this is the case post 2 doses, 8 months out, that the vaccine effectiveness against severe disease remained above 95%, and against infection, it declined to about 71%. And also within 7 days of a 3rd dose, recognizing that there was antibody waning, VE against infection boosted back up to over 90%. And then in the context of the 3rd dose with Omicron, the extended analysis to date in the test negative design suggests that we have over 95% protection from severe disease and about 60% protection from symptomatic infection. Next. So then the data also very encouraging that 3rd dose boosters have helped to improve immune protection, especially for those at risks. And we saw that all parameters of immunity in the SOTR, solid organ transplant recipients across the alpha, beta, and delta variants was improved with the booster. Similarly, we saw that booster doses provided better neutralization against variants in the long-term care residents, thanks to Dr. Bowdish and Costa. And then we also noted that boosters are required for all adults 7 months past dose 2, as Dr. Kwong's study suggested, is that as the interval after the 2nd dose is extended, your protection from infection declines, and therefore to protect against infection in the general population, the benefit of the 3rd dose. Next. Factors to consider on the dose interval. Protection from risk is from infection risk higher in the healthcare workers who had a longer dose interval compared to healthcare workers who had a shorter interval. And this was very clearly showed by Dr. Ferreira, although Dr. Kwong's study suggests that that may not be as important in understanding vaccine effectiveness compared to the dose time since the second dose, which is the next finding. And here we see across all patients that the time since that second dose is directly associated with a decline in protection from infection. And we also had interesting evidence that the vaccine type matters, that antibody quality and quantity was higher and longer, and memory related to the cellular immunity was higher amongst long-term care residents who received Moderna versus Pfizer. Next. So these are amongst the things which we had findings where we saw that these studies do matter in terms of policy. We are promoting universal vaccination, and research and science backs that very fundamentally. Booster doses for all adults, beginning with those at greater risk, has also been clearly demonstrated through this science. And we have the Omicron wave, which not only makes it clear why universal vaccination and boosters have been important, but has led us to recognize that we need to look at transmission in the setting of high levels of vaccination, and recognize that other public health measures related to decreasing risk of infection and transmission cannot be overlooked in this setting as well. Next. So there's still a lot of questions out there, and I think many of them were referred to, and there are many in the chat, and we'll be moving to this in a second, but we have lots of questions whether and when boosters should be recommended for children and teens. Not a focus of today's talk, but one where with the rollout of vaccinations in that population starting several months ago, there will be lots of work on that front. And then also whether, when, and for whom additional regular boosting will be required once SARS-CoV-2 becomes endemic, as it appears to be headed. The CITF studies, these and others, will be looking at these questions, and we will endeavor to keep the public and policymakers updated as data emerges. Thanks very much for your attention.
Thank you very much, Dr. Evans, for that summary. And I think we are going to be moving to a gallery view quickly, and we will take questions from the members of our audience. If you haven't really written any questions, we have lots of questions. We're not going to get through all of them, so we are trying to arrange a way that we will get we'll get answers to your questions and maybe post on the website and so on, and we'll be communicating that a little bit later. Let's begin with a couple of questions for Dr. Jeff Kwong. And I just need to get to the questions, Jeff. So the first question to Dr. Kwong: are your conclusions today still that VE against infection caused by Omicron is 37% after third dose? If so, does this raise the discussion regarding whether a fourth dose makes sense, or whether there should just be a retooling of the current version of the vaccines to make them Omicron specific? So let's actually go with that first question first.
Yeah. So, I think I showed today that basically, the VE depends on the outcome that you're using. And I think the most important outcome is the severe outcome, so hospitalization or infection, or death. And you can see that it's very high VE after the third dose. And still, there's still some protection even for those who've had 2 doses only against Omicron. Against symptomatic infection, you can see that VE drops to basically 0 by 6 months after your second dose against Omicron. But it does go back up to 60% or so after the third dose. And so I think, I think that's worthwhile, getting some protection, even if it's not 90-something percent, 60-something percent is still worthwhile. Certainly higher than the 37% that we initially saw. and which we attribute that because of a combination of biases, because of the outcome that we were using any infection, and like who's getting tested, as well as the very early period when Omicron was first circulating, leading to bias in terms of like who's getting exposed to Omicron. So I think definitely we should be going for third doses. I would question the need for a fourth dose for the general population. I think Don and Victor made very good arguments why for very high risk populations, a fourth dose makes sense, right? Because I think a fourth dose in those who have immunosenescence or immunosuppression is equivalent to the third dose for those who are immunocompetent. So basically the short answer is I would not recommend a fourth dose broadly.
Thank you very much, Dr. Kwong. And we have questions for all panelists. So, but we will kind of go through in these questions one by one. Jeff, I still want to ask you your second question as well, I mean, if you don't mind. And that is, could you expand on this negative effectiveness, reasons for negative effectiveness? I think a lot of people are interested in this observation from your team, and particularly versus Omicron infection for that 2-dose regime, even in less than 2 months after the second dose, VE was nearly zero, like no efficacy at all. Which seems unlikely. So, Jeff, over to you again.
Yeah, so I think I just want to stress that these are observational studies that we're doing, and they're not randomized controlled trials. So there are differences between people who are vaccinated and people who are unvaccinated that sometimes that can't be controlled for in the analyses. And that's what we call residual confounding. And so we try our best by controlling for as many factors that we're able to measure, and we can account for them in our statistical models, but there are things that we can't account for. And so, as I've mentioned, earlier, one of the problems with the early, when Omicron was first introduced in Ontario, it was people who were traveling, who were getting infected and bringing it back. They were infecting their household members and their social contacts. And so people who are traveling were all double vaccinated. You had to be double vaccinated in order to travel, right? And then, probably these cluster in households and social networks. So people who were contacts of these double vaccinated travelers were also probably likely to be double vaccinated. So early on, people who were double vaccinated had much higher exposure risk to Omicron than people who were unvaccinated. Because of the vaccine certificates, people who are unvaccinated were not allowed to go to restaurants and go to the Raptors game or the hockey games and be exposed to Omicron. And so that's why it looks like there's a negative vaccine effectiveness, that people who are vaccinated had much higher exposure to Omicron than those who are not vaccinated. And then there's I think we saw that particularly when we looked at infection as the outcome, because we think that what's happening is that people who are vaccinated and exposed are more likely to go access testing than those who are unvaccinated. And then that will further cause, make the VE estimates go down, and then giving like a negative VE estimate. But, I think our updated analyses show that, if you're just careful about the outcome measure and, consider all of these things, then you can see that the VE estimates are not truly negative, but they do suggest that they do go to zero after a certain amount of time after the second dose. And that speaks to the immune evasion of the Omicron variant compared to previous variants.
Thank you, Jeff. Yeah, really interesting. I'm going to go to Dr. Bowdish and Dr. Costa now, but I will just say this, Jeff, and maybe if we have a little bit of time at the very end, I'll come back to it. And that is outcome. So is it prudent to be looking at infection in general as an outcome, or should we really be moving towards those serious COVID-19 outcomes, hospitalizations, deaths, and so on? So, and so I'll just park it there, Jeff, for you to think about. And I want to come to Dr. Bowdish and Dr. Costa. The question is, your data would suggest a fourth dose at 3 months. Is this the right interval? Is this strategy going to be applied to recommend fifth and sixth doses as well in your population? Does this fourth dose strategy extend beyond long-term care and retirement home populations? So can you generalize it outside of that population that you did your studies in?
Andrew, do you want to weigh in, or shall I? Andrew and I can finish each other's sentences, so we're kind of interchangeable.
You start, absolutely. Go ahead.
Okay. So this population is a special population. I am not in the least bit worried about myself needing a fourth dose. At this point, I'm very happy to wait for Jeff to tell me when the time comes, when there are more breakthrough infections, serious outcomes. Our population doesn't seem to be able to hold on to the immune response quite as long as a younger, healthier population. So this fourth dose is a mixture of pragmatism and also immunology. The pragmatism aspect is once these infections get in the homes, they are incredibly debilitating. We do not have really good options for treating people with complex comorbidities, and we just don't have the capacity. So a fourth dose was and is an investment at dealing and helping us all get through this Omicron wave so that our frail older adults who've already suffered so much because of these infections don't end up in the hospital. So there's a pragmatic component to it. Time will tell when that fifth and sixth dose is necessary. And I have to say that one of the things that will also influence this is community rates of Omicron. Our long-term care residents have the infections come into the homes and become infected. So when those rates are lower, we have fewer infections in general. So time will tell. Groups like ours and others will continue monitoring these, but I do not think this applies to the general population. The surprising feature for me, frankly, was that our retirement home communities were on the same trajectory. I honestly thought that our frail long-term care residents would be more immune impaired than older but healthier population. But the fact that they seem to be on the same trajectory tells us that a fourth dose might be right for them.
Thank you very much, Don. Andrew, do you want to add anything to that at all?
Nazeem, I might take a prerogative and answer another question there that was, well, why not other frail older adults living in the community?
Yes, please.
It's an important calculation. What factors in is the fact that for older adults living in long-term care or other congregate settings, they're higher risk for exposure, and so that changes the calculation around risk. With Omicron, that changes that original calculation slightly because everybody's at high risk of exposure. But the difference for those in congregate settings is that staffing is so frayed that an infection means disruption to care to a greater extent than it does for older adults at a similar frailty level living in the community. And that's the pragmatism that Don mentioned.
Thank you very much, Andrew. Thanks for that addition. And I have a question for Dr. Ferreira, and as you see that there, given the percentage of SOTR having positive antibodies after third dose, especially against Omicron, should the 6-month interval for their booster dose be reconsidered? So it's a dosing interval question for you, Victor.
This is a very good question. I think the evidence that we're seeing so far does show that, at least with respect to first and second doses, the rate at which antibodies sort of contract or decline might actually be slightly faster than what you would see for the general public. And we've kind of been using the 6-month duration for a while there to assess the kind of the time point for when boosters might be necessary. But I think especially in light of the Omicron variant and the fact that we see triple vaccinated transplant patients being admitted to hospital, that 6 months might be too long. And unfortunately, most of the data that we are seeing with fourth doses are looking at kind of a smaller interval between their third and fourth doses. So the studies so far have been mostly focusing on 1 or 2 months post the third dose. But the good news in Canada is, although NACI recommends a booster, a fourth dose, at least 6 months after the third dose, in some provinces like Ontario, Saskatchewan, and Alberta, you can actually go a little bit earlier than that. So in my opinion, I think 6 months might be too long, especially in the context of the current, in terms of Omicron and the increased infectiousness, and the fact that so many transplant patients, even with 3 doses, are ending up being hospitalized.
Thank you very much, Victor. We have about 5 minutes according to my clock here, and I think, but Jeff, I'll come back to you to perhaps weigh in on that question about what's the outcome of real relevance right now, Omicron and post-Omicron, in relation to vaccine efficacy. How should we even be communicating our vaccine efficacy results on studies, infection, symptomatic, serious COVID-19, etc.?
And I think the severe outcomes is the most important outcome. I think we can all agree on that. I think the challenge is that, if you allow more transmission to occur, then there's, you will get more severe outcomes just by sheer force of infection. So just by having more infections, you're going to get more severe outcomes. So I think, to me, the bottom line is, it's good with 3 doses, we have high protection against severe outcomes. I think we can't rely only on the vaccines. As Tim alluded to in his comments, we really need the other non-pharmaceutical interventions. We need to continue masking. We need to continue, limiting our social contacts. All of these things, improving ventilation. All of these things are what we need. We can't think that vaccines alone are going to get us out of this pandemic.
Thank you, Jeff. That does my heart well as a public health person. That it's not just one thing. Even vaccines, even including vaccines. Vaccines is not the panacea, and I think we kind of come to that conclusion pretty quickly here since late last fall, when Omicron reared its head. So thank you very much. We have literally 2 minutes, and in that 2 minutes, I want to make some quick announcements. First, I want to let people know that your answers, question to answers, will be posted on CITF website in the coming weeks. I'm told that in a chat box, and also the newsletter that you will receive will give you the heads up, alert you to go to the website and read the answers that you have provided, the questions that you have provided. So that's number one. Number two, I can't thank enough the panelists. This is really, really timely work, and as Canadians, we should be proud that we are doing this work right here in Canada. And thank you very much for those who present. And we do know that you represent a whole team behind you. So thank you very much. The next event, that's number two. Now number three is next event in this series will be in late February. So stay tuned for more information on that. And if you are interested in seeing any of the publications referenced in today's event, you can find them listed on CITF website, the same website where the answers will be held at the end of the event, as well as a recording. So thanks again for joining us, and stay safe. Take care, everyone.
We would like to thank today's speakers and attendees for their time, as well as the members of our networks for their continued support and participation in Canada's pandemic response. If you are interested in attending our monthly research results and implications events, please visit either website for more information.